Hi, BISC 210 students. Today we're going to cover Chapter 9, Infectious Disease and Epidemiology. Now with this chapter, it is a great segue into starting to move towards our second half of the semester information regarding diseases of the organ systems and case studies. This is kind of a little bit of a bridge of what we have learned and what we're going to be learning. With this chapter, I will admit there is a lot of dense information, meaning a lot of definitions which you have to know. There's a lot of thinking outside the box, so to speak, when it comes to epidemiological studies. So this chapter kind of has it all, but I will make sure that you're aware that there's quite a few definitions. So we'll see that as we go through the chapter, and this is a really great chapter, and it applies to what's going on in the world today. In the world of COVID, infectious disease and epidemiology are important. It's important that we understand this information, because next time you're watching a show, TV, a news program, and they start talking about epidemiology and all these fancy things when it comes to COVID, you'll be able to understand it and follow along. So let's start this off by just simply talking about terminology. Infectious disease basically just means that there is a pathogen that will cause illness. Now, we've talked about numerous pathogens examples of pathogens that can cause disease. It's not just bacteria and viruses, right? We have a whole list of types of microorganisms. So those pathogens, the organisms themselves, can cause illness. Now, not every single one will cause illness, but some will. Remember, with bacteria, only about 2 to 5% of the known population of bacteria is infectious, can cause disease. So it's a small portion. And we have listed here the types of pathogens, which should come as no surprise to you. Viruses, bacteria, protozoans, fungi, helminths, prions, which we're not going to talk about in this class. Basically, those are misfolded proteins. Prions caused mad cow disease. Basically, a messed up protein touches another protein, and it changes it and makes the next one not functional. And then the next one, it's a chain reaction. Anyway, that's about as far as I'm going to go into prions in this entire class. And then the term epidemiology. Basically, epidemiology is the study of controlling, monitoring disease globally, and then also promotion of public health. The CDC, for instance, the Centers for Disease Control, has this huge epidemiological branch where they monitor emerging diseases, which we'll talk about what an emerging disease is later on. Or they monitor the spread of a disease, right? So epidemiologists right now they have their hands full with the COVID problem. By the way, COVID-19 is a short term, is a name that the virus was given, just to have kind of a shorthand way of saying it. The actual virus itself is SARS-CoV-2. So SARS, S-A-R-S hyphen C-O-V hyphen 2, SARS-CoV-2. So it's important to know that monitoring these types of diseases and disease occurrence is really useful in terms of future control of that disease. For instance, the flu virus. There's over 200 versions of the flu, but epidemiologists every year, they monitor which ones are the most common. They monitor which ones in Europe are more common because their flu season's a little bit before ours. And then that's how we build our vaccines against only 3 to 4 of the most common flu viruses of that year. So epidemiologists have a really important role in all of this tracking and monitoring and then coming up with solutions. And really, in the end, it comes down to promoting public health. Like the CDC sets out these recommendations for vaccination schedule. For COVID, they set social distancing regulations. They set mask regulations. They put into place numerous aspects of ways to help control the spread of the disease and promote public health. Now, is that being followed by many people? No, which is why the United States leads the world in COVID deaths. We'll get into that a little bit later. There are two distinctions between pathogens. There are opportunistic and true pathogens, and their definitions are pretty much what you would expect. Opportunistic pathogens will cause disease when the host is weakened. So let's say you have someone who is undergoing cancer treatment. They are undergoing radiation and chemotherapy. Their immune system is weak from the radiation and chemotherapy. So they are at a higher risk for opportunistic pathogens to cause disease. They're also at higher risk for true pathogens. But a healthy individual most likely wouldn't be affected by many, I don't want to say all, but many of these opportunistic pathogens because we're healthy. But if our immune system is weakened or suppressed in some form, they can take advantage of that. A really good example is on your skin, you have a bacteria called Pseudomonas aeruginosa, and it's an opportunistic pathogen. It hangs out on your skin, it just lives there, and it doesn't bother you at all until you get a cut. Wherever you get a cut, let's say on your arm, your forearm, if Pseudomonas aeruginosa is present, it can potentially cause an infection. So it will take the opportunity to cause infections. There's a lot of other situations like this. For instance, women have a really unique balance of organisms in the vagina. And when a woman gets treated for a urinary tract infection, given antibiotics, antibiotics don't discriminate between pathogen and non-pathogen. They just see something that they're supposed to attack and they attack it. So, a lot of women actually end up with yeast infections after or while they're getting treated for a urinary tract infection, because the antibiotic starts to weaken the normal microbiota, the number of bacteria in the vagina that controls yeast. It prevents yeast from actually colonizing and causing a yeast infection. But if the woman is being treated for a UTI, for instance, a urinary tract infection, and she's taking antibiotics, then that those normal bacteria are going to drop in numbers, and then all of a sudden the yeast can take over and cause the yeast infection. It's an opportunistic pathogen. True pathogen is just one that does not need a weakened host at all. It can just get you sick, period. You don't need to be weakened. You can be healthy as a horse, as they say, and still get sick with this pathogen. Some bacterial examples, Shigella. Shigella is one, Salmonella is another, Yersinia is another. There's quite a few. Giardia, which is a flagellated protozoan. But anyway, the point is they don't need you to be, your immune system to be weak. They can just attack and cause illness. And in this class, when we get to the diseases of the organ systems and start going one by one organ systems, we will talk about these true pathogens. There are some that are opportunistic, but there are quite a few that are just true pathogens. That you come in contact with it, you're probably going to get sick. Now, these four terms are really, 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 really important. Okay, sporadic. This is in reference to cases and of a disease and the spread. So sporadic is simply isolated infections in a population. So let's say in the state of Mississippi, we get, and we'll use Ebola, 4 cases in Lafayette County, 4 cases in any other county in the surrounding area, 2 cases some in a different county. Those are sporadic. They're basically within the population of the state of Mississippi. There's some random isolated infections throughout the state. Now, the term endemic means that disease is regularly found in a population or region. Now, this is saying head colds. Head colds are an okay example. I think a better example would be malaria. Malaria is a disease that's passed via mosquitoes, and what happens is basically malaria, although it is not found in most of the world, there are certain regions of Africa where you will absolutely find malaria to be extremely common. And that's what an endemic infection is. When you have an infection that's commonly found in a population or region. So with malaria, if you look at certain parts of Africa, and you can say, well, in this part of Africa, say West Africa, there's malaria present in that region pretty much at all times. It doesn't really go away, come back, go away, come back. It's just there. And it doesn't really go away. Then you get epidemic and then pandemic. And then these two terms are really close in terms of what they mean. So epidemic is a widespread disease in a specific region during a specific time frame. So when COVID first started, say in January-ish, it was an epidemic in Asia, right? Mostly in China, a couple of the surrounding countries near China. So it was spread out, it was widespread in a given region, and then we could say over the, January through March, it was majority of the time in that area. it hadn't really spread to the rest of the world much. I'm sure there were cases elsewhere, but for the most part, the disease was kind of isolated to that area, but it was really widespread between a given amount of time. When it starts to move towards a pandemic, which is essentially a worldwide infection, that's when you start to see significant transmission from one region of the globe to another. So, for instance, if I were to say COVID-19 started in China in January and was extremely prevalent from January to March in Asia, or you could say Southeast Asia, to be more specific. That would be more of an epidemic. But if I were to say COVID started in Southeast Asia in roughly January, and then by March, most of Southeast Asia was seeing large outbreaks, and then by May, it had spread to 87 other countries. Okay, that's when you start having to look at it as a pandemic, as opposed to an epidemic. Okay, we're looking at widespread in terms of pandemic, widespread throughout the globe, not just one particular region. We're talking about the entirety of the globe. And that's what we're in right now, is a pandemic. Because if you were to look at a distribution of COVID, you're going to see that it has spread to almost every single country on the planet. There are a few that have not really seen cases, or they may have seen cases, and those cases are because the person traveled to another country and brought it back. But the point is if it spreads to 87, 100 countries, then you're starting to consider it a pandemic. Another example of a pandemic was the bubonic plague, or the Black Plague. It was spread through ticks, and people say it was. So when you read about the bubonic plague, most people associate it with mice, but it's not actually the mouse that was causing the illness. It was the tick on the mouse that was infected. But the point is that plague spread to numerous countries across the globe. Now, granted, back then they didn't have completely defined countries as we do now, but the point still remains that it was extremely widespread and wasn't restricted to a particular region and a particular time. That's what sets it apart from an epidemic. Emerging versus re-emerging. Emerging simply means new. Re-emerging means it's coming back. So COVID is an emerging pathogen. Zika, Ebola. But perfect example is COVID. There are lots of SARS versions of SARS. But SARS-CoV-2, which is what causes COVID, is something we have never seen before, which is why it's so dangerous. Because when you see something for the first time, there's no way to, be prepared, so to speak, for it. right? It just shows up and you're like, whoa, wait a second. I've never seen this before. How do I go about treating it, fixing it, getting rid of it, right? And that's the problem we've run into here in the United States and globally, is SARS-CoV-2 just spread really fast and we were not ready. And since it had never been seen before, we didn't have a vaccine, we didn't have a treatment plan, we didn't have, individuals didn't have immunity to it. So you can see that new emerging pathogens are extremely dangerous. And that's why SARS-CoV-2 or COVID-19 is so dangerous. Now there are re-emerging pathogens as well. Basically, we got them under control, but now they're resurfacing. Antibiotic resistance bacteria is a great one. For instance, Staphylococcus aureus, Staph aureus, is a pathogen that causes numerous diseases in humans. But MRSA, which is methicillin-resistant Staph aureus, actually has the resistant gene to all the cillins, penicillin, ampicillin, amoxicillin, all that stuff. So they can resurface as resistant strains. Another really good example of re-emerging pathogen, which is unfortunate, it's measles. Measles is something that we have a vaccine for, and for a very long time it was what we considered eradicated in the United States. There's less than, I think it has to be less than a couple hundred cases per year. I want to say less than 100. I'd have to look up that exact number, don't quote me on that. But it's under a certain number of cases per year, we consider it eradicated. And measles was one of them. We have an effective vaccine. We're able to, people get the measles vaccine and protection. Boom, perfect. But with the anti-vaccination movement that's going on right now, we are now starting to see the reemergence of measles. 3 years ago or so, 3 or 4 years ago, when this first started happening in terms of measles outbreaks, there was an outbreak in Disney whatever in California. I can't remember if it's Land or World, whichever one. The Disney in California, there was an outbreak of like 300 something people that got infected with measles. And then there was another place, there was actually an NHL hockey team that most of their players ended up with measles. So the point is this pathogen came back because we as a species, humans as a species, just kind of got lazy about it. And something that's totally preventable, ended up becoming a huge problem again. And it still is, even though we have a vaccine for it. We'll talk about the anti-vaccination movement a little bit when we talk about vaccines. So some more terminology. Communicable disease is a disease that can go from human to human. So for instance, COVID-19, the cold, the flu, staph infections. I could go on and on and on and on. From human to human. Any STI or sexually transmitted infection or STD, sexually transmitted disease. There is a difference between the two. And when we get to the reproductive system, we'll talk about STI versus STD, why we differentiate. There are non-communicable diseases. That would be something like cancer, for instance. It's a disease, but I can't transfer, if I were to have cancer, from myself to another person. Okay? So there are some diseases that just can't be transferred. And For instance, a non-communicable disease would be cancer. Contagious diseases are diseases that are easily transmitted from one host to the next. This is a really hard definition because the term easily is subjective, right? You may say it's easily transmitted in X amount of time, and I may say, well, I think it takes a little bit longer, so I'm going to say Y amount of time. Who's right? Well, without research, we can't answer that. So it's really hard to say something is easily transmitted from one host to the next, because of the fact that in some cases we just don't know. We just don't know how easily transmitted they are. However, in our current situation with COVID-19, we do know how easily a disease can be transmitted from one person to another. We know something called the R0 value, or RO value, and I'm going to talk about that in a few minutes. So we'll talk about how contagious diseases are in a few minutes. And then the term zoonotic means disease goes from animals to humans. Good example, rabies. An animal that's wild, that has rabies, bites you, transmits the virus, you get sick. That's a zoonotic disease. I have a story about rabies when we get to that later on about my wife. About my wife and a fox. All right, so again, as I mentioned in the beginning, this is kind of definition dense. So I'm sorry. I wish I could make it a bit more fun, but unfortunately, this is the way we have to kind of go about it. Now, the next terms I want to talk about are the terms signs and symptoms. This is something that almost everybody uses incorrectly. And it's important that we kind of clarify what we describe signs and symptoms to be. Signs are things you can measure. For instance, temperature, right? Patient comes in, say, I don't feel well. All right, that doesn't help much, but we can figure out what are the signs associated with that person not feeling well. So we say, all right, the signs. We take their temperature. They have a fever. We take their blood pressure. Their blood pressure's up. We take their heart rate, their oxygen saturation. And we can quantify these things. So signs are things you can quantify. Symptoms are simply how somebody feels. So I go in and I say, my stomach doesn't feel good, or I feel achy, I feel tired, I feel nauseous. These are things that the person experiencing them is the only one that can really can identify and describe. Whereas quantifiable information like signs, blood pressure, temperature, can be seen by the primary care physician or nurse practitioner. And then what we do is we then combine the two, and by combining them, we can kind of figure out an overall picture of how this person's overall health is. So at that given moment, not overall health in terms of are they healthy in terms of their life overall for every day, all day, but more specifically that given snapshot. So when next time you go to the doctor, keep in mind when they start taking your temperature, when they start blood pressure, oxygen saturation. If they put an EKG on and look at your heart rate, or heart electrical conduction. All that stuff is used to quantify. Even with pain, we try to quantify pain as well. There's a scale from 0 to 10, so we even try to quantify pain, because technically pain is a feeling, right? How much pain I can handle, my threshold for pain, is probably very different from somebody else's. So by trying to quantify it, we can help standardize the concept of pain and then know how to treat appropriately. If somebody has a pain level of 2 and they say, all right, I have a pain level of 2. That does not warrant you giving them morphine. Right? It does not warrant you giving them morphine. In most cases. Now, for somebody else, that 2, that maybe I felt a 2, maybe that feels like an 8 for somebody else. And based on all the other signs and symptoms, that 8 says, all right, this warrants the patient getting morphine. So the point is quantifying allows us to help standardize a little bit. Now, I'm not saying I'm a total badass and I have, a pain threshold of like 2, and I'm like, yeah, nothing hurts. And then everybody else is like, oh yeah, his 2 is my 8. That's just an example. Everybody is different. You could give 10 different people the exact same symptom and of pain and ask them to describe what the number is and get a handful of different answers. So it's subjective, but we try to quantify it and make it standardized. There are 3 major types of diseases. There are acute diseases, which happen very quickly, meaning you get sick, you get infected and sick really, really fast. Infected and sick really, really fast, and then the symptoms, the signs and symptoms appear rapidly. Chronic diseases or persistent diseases have slower onset and progression. And then latent diseases have random symptoms and progression. So we'll talk about some examples of each of these. Oops. Sorry. Acute would be something like Salmonella or Shigella that causes food poisoning or food infection. Let me clarify food infection. There is a difference between food poisoning and food infection, which we will clarify when we get to the GI system. But the point is, a food infection or food intoxication, that it happens rapidly. You eat food, 12 hours later, you're puking and you're throwing up and you have diarrhea and you're nauseous and all these other things, all these signs and symptoms. That is a rapid onset of symptoms and signs, and you're sick very quickly. And then the progression happens quick, and then you get a fast drop off afterwards. Chronic is something that slowly builds over a longer period of time and then expresses itself, and you are sick for however long. So a good example would be something like HIV. HIV, when you're infected with this, it slowly builds the virus over years, potentially, and then at one point it becomes active and very prevalent, and basically at that point it becomes AIDS. And so the progression is more gradual. Okay? You may still feel some signs and symptoms early on, but not very significant ones. You may just feel a little tired, nauseous, and just kind of a little meh. But as the time goes by, it starts to get worse and worse and worse and worse and worse and worse. Latent diseases are random, and the symptoms show up random, just literally randomly. A great example of this would be human herpes virus 1 and human herpes virus 2. Basically, cold sores on the mouth and cold sores on the genitals, sores on the genitals. So human herpes virus is called HHV1, is caused by the herpes virus, and it causes cold sores. Now, what's interesting is according to the CDC and a bunch of research, about 60 to 70% of the population of the U.S. has or has come in contact with human herpes virus 1. But what's interesting about diseases like that, latent diseases, is you may have it, but you may never, ever, ever see it. It may never express itself. So let's say you get infected with HHV1, human herpes virus 1, which causes cold sores. You get infected, and you go the rest of your life never having a cold sore. And you don't even know that you have the virus. You just do. That would be considered kind of latent. It's hiding. It doesn't show up. But randomly, 5 years later, 10 years later, you get your first cold sore. Even though you've had it for years before that, you get your first cold sore. And then after that cold sore has healed, 2 weeks or so, depends. 2 weeks is usually around the amount of time it takes. It heals, and then it goes back into hiding for however long. Another 5 years, 10 years, or may never come back again. So it's kind of random. In terms of human herpes virus 1, we kind of have an idea of when it expresses itself. We don't know for sure, but we're pretty sure that if somebody has human herpes virus 1 and their immune system is weakened and they're stressed and they're unhealthy, then there's a higher chance of the virus being expressed, as opposed to someone who's infected who is extremely healthy, their immune system is strong, and they have healthy diet, and overall their health is better. So we don't know for an exact reason why it goes away, comes back at random intervals, but it does, and that would be considered latent. And we'll talk about several diseases that are latent. Koch's postulates. Hopefully you remember these from earlier in the semester, but I will review them. First, the same organism must be present in every case of the disease. The organism must be isolated from the diseased host, the sick host, and grown in a pure culture. Now, when we talked about this in chapter 1, I believe, we mentioned this term pure culture, but we really didn't describe it or know what it meant. Now you should know what it means to get a pure culture, right? Streaking for isolation, right? We grow it as a pure culture. We streak for isolation. Then, the isolated organism should be introduced to a healthy host, and it should cause disease. It should cause the same disease that you saw originally. Then in the last one, organism must then be re-isolated from the sick disease, the inoculated or diseased animal. So the one that you just introduced the pathogen to, if it gets sick, then you have to re-isolate it. When I was at Mississippi State, we worked with ticks and mice, and we looked at transmission rates of a bacteria from a tick to a mouse. And we would take mice that were healthy, we would attach a tick to them, and then we would let the tick bite them for X amount of time. We'd remove the tick, and then we would take blood samples from the mice afterward. Now, not right away. We would give them a little bit, give the mouse, allow its body to build up some antibodies against it and mount an immune response. But we would look at blood samples and try to figure out, quantify how much of the pathogen was transmitted. So you have to have a healthy host that you can re-isolate from. Now, there are limitations to Koch's postulates, and there's some major ones. First, non-infectious diseases don't apply to this. Right? Non-infectious diseases don't apply to this. So infectious means it could be transmitted. We said cancers can't be transmitted, therefore this doesn't apply to cancer. Certain diseases cannot be isolated. So certain pathogens can only be present in vivo, which is in life, and not in vitro, which is in the lab, meaning that you could try to grow it in a lab, but it won't work. Some organisms can't even be isolated. Some organisms are so dependent on hosts that if you try to isolate them, it doesn't work. So certain diseases can't be isolated in a lab. And again, here are two examples. As I mentioned, they can't grow in a lab. And then obligate intracellular pathogens, meaning pathogens that have to live inside human cells. So there are pathogens that have to live inside of our cells to cause disease. The second you take them out of our living cells, they die, because our cells will be dead, and they can't get the nutrition and whatever they need from those cells. So sometimes it's almost impossible to get some intracellular pathogens to grow in the lab. In pure cultures, some microbes become attenuated. Attenuated just simply means broken down. Some microbes don't infect humans. There are some bacteria out there that don't infect humans. They infect animals, but they don't infect humans. And then the other way you can take this statement is, so some diseases are not every disease is a human disease, is the way what I was describing. The other way you can take this is, if you have a human disease and you want to learn more about it, we don't like to run studies on humans, right? It seems to be a bit unethical. So what we mostly do is we use animal models like mice. We use mice or primates, or several other types of what we call animal models. And sometimes some pathogens are just human diseases. They don't infect non-humans. So there's two ways you can kind of take that that one right there, that last one. So where do pathogens come from? They come from reservoirs, which are animate or inanimate habitats where the reservoir or where the pathogen is naturally found. For instance, the reservoir for Clostridium tetani is typically the soil. A flagellated protozoa called Giardia. Its habitat is typically stagnant water. Okay, so you can kind of see those are two inanimate examples. Animate examples would be Yersinia enterocolitica, which causes gastrointestinal disease, mostly kind of like food poisoning-ish. It's found in pigs. Or bearded dragons. So my pet bearded dragons, all three of them, they have salmonella on their skin as part of their normal microbiota. To them, it's normal. Salmonella to us is pathogenic, which is why we have to wash our hands immediately after holding any of the three dragons. Or letting our son touch, pet the dragons. He has to wash his hands immediately because introducing that salmonella into his body could risk infection. Then you have what we call endogenous and exogenous. Basically, endogenous means the pathogen came from your own body. Exogenous means it came from somewhere else, external. I mentioned Pseudomonas aeruginosa being an opportunistic pathogen. That would be an endogenous source. If it lives on your skin and it's there all the time and then you get a cut and it infects, makes sense. Exogenous would be you eat a undercooked steak or raw burger and you get sick with salmonella or Shigella or any GI pathogen. And when I say GI, I mean gastrointestinal tract, which is your stomach, small intestine, large intestine, colon, all of that. Actually, you can even include your mouth in that as well. Okay, so different sources, some of which are reservoirs. This table is useful. It is in your book. It's table 9.2. Talks about exogenous versus endogenous. And I literally just covered environmental and then misplaced normal microbiota, which are opportunistic pathogens. So you can take the time to read these last few ones. Transmission. So how does the pathogen spread from host to host? Two major ways are direct contact and indirect contact. And there's a bunch of subgroups for each of these. And this is figure 9.1 in your book. So on the left, we have direct contact, person to person, animal, environmental, vertical from mother to child, or indirect, airborne, vehicle, vector, biological vector, and then mechanical vector. Now, we'll talk about each of these. Okay. Direct means you have direct contact between the source of pathogen and host. Indirect means there is no direct contact between the source of the pathogen and the host. Okay, so there's no direct contact. Now, we're going to talk about this because it's relevant. Well, not only is it important in general, but it's also really relevant to what we're dealing with now with COVID. So, direct contact, the host comes in physical contact with the source of the pathogen. For instance, a bite from a rabid dog that has an animal that has rabies. Kissing, swimming in a pond. I honestly, the definition of taking a dip in a pond comes from the book, and I actually hate that. Going for a swim in a pond. Let's do that. Or in a lake. It doesn't really matter. There's pathogens in all of that. In all of them. And then indirect, there's no physical contact with the source. And then there are three major categories: airborne, vehicle, and vector. Airborne, vehicle, and vector. And all of these are indirect contact, which leads to transmission. And we're going to talk about each of them. Okay, another second or so. Let's write this down. Airborne, the pathogen enters through the respiratory route through droplets. Now, airborne can actually be a little bit confusing because some pathogens are airborne in the sense that they hang out in the air for a long period of time. Others are airborne in the sense that they hitch a ride on any saliva that leaves your mouth, from coughing, from laughing, from sneezing, from breathing, or any of that. You're expelling respiratory droplets, and on those droplets, the virus or bacteria hitch a ride. And the big difference is the ones that are airborne, that are not respiratory droplets, stay in the air a long time. Tuberculosis, for instance, can hang out in the air for extended periods of time. So if you're in the airport and you're standing there and somebody has tuberculosis and they're coughing and coughing and coughing, and they walk away and then you take their spot in line, well, chances are there's probably still some of that pathogen in the air. Respiratory droplets, on the other hand, only travel a certain distance and then they fall to the ground because they're heavy. Vehicle transmission, basically the pathogen is on a fomite. So a fomite is an inanimate object. So a fork, a spoon, towels, syringes. So the pathogen is found on a contaminated fomite, which is an inanimate object. Inanimate object. And then vector transmission, you have typically arthropods like ticks or fleas, for instance, that are hosts to the pathogen, and then the pathogen can be transmitted. This is what I was talking about with the bubonic plague. The mice that were infected were not infected because they had the virus per se. They didn't just, magically get the virus. It's the ticks or the fleas, excuse me, not ticks, fleas that were biting them that transmitted the virus. And it's very interesting because even back then, when the bubonic plague was kind of really predominantly active, there were certain places that were at war, certain countries that were at war, and they would actually take catapults and put mice in them, dead mice, that they figured were infected, and they launched them into cities, over the castle walls and into cities, to try to infect them and get them sick to the point where the population would drop and they would die. That was some of the early signs of biological warfare, which is pretty crazy, that they figured out, oh yeah, this mouse is infected with something, let's throw it over the wall and get somebody else sick and they'll die, and then eventually we can take over the place. Very weird. Also very smart, in my opinion. Wrong, ethically wrong, but kind of smart. Alright, so in terms of vectors, there are two major differences. There's biological and then mechanical. Biological is when the organism has a role in the pathogen's life cycle. So for instance, when you're talking about parasitic worms, the larval form is microscopic and it's part of the vector has a role in the pathogen's life cycle. Ticks and mosquitoes, for instance, when they have diseases, the larval forms of the diseases are found with the organism having a role in that life cycle. Now, mechanical vector, on the other hand, is just simply something that hitches a ride, so to speak. So it's not part of the vector spreads the disease without having it being part of the pathogen's life cycle. For instance, flies. So flies, you're at a picnic, they fly around, they land on one of the trash cans, they land on some of the trash that's been there for days, old, dirty food. They land, they eat something, and then they get up and they start flying again, and they fly over to your picnic, and they land on your burger bun. Okay, they land on the bun. And you shoo it away, and then you eat your burger. Well, what has happened in that case is that vector has actually transported and transmitted some potential pathogen from its feet to your burger bun. So let's say it lands on the trash, picks up a pathogen on its feet, flies, lands on your burger bun. Wherever its feet land, it's going to have some transfer of that microorganism. Some transfer of that microorganism. Which is interesting. Now, granted, the likelihood of you getting sick is pretty low because of the fact that the sheer numbers of the pathogen are really low. So when you have mechanical transmission, the fly landing on your burger bun, maybe it transfers 100 cells, 1,000 cells. Depending upon what pathogen it is, you may need hundreds of thousands of cells. Or some pathogens you need less than 10. Shigella, for instance, which causes a GI disorder, requires less than 10 cells to cause illness. So, depends upon the organism that they pick up, but in most cases, if you're healthy, you really don't have much to worry about in terms of a fly landing on your burger and you shooing it away and then eating it. You're fine. Some key terms here. So before we move into some more definitions, we've already seen this slide. I just want to reiterate some of this info. Okay? Direct contact, person to person, animal, environmental, vertical. Indirect, no direct contact, airborne, vehicle, vector, biological vector, and then vector mechanical. With COVID-19, this right here is why we wear masks. Viruses have to live in host cells, and if we cough, then we're going to allow them to latch onto the respiratory droplets and be transmitted to somebody else who comes in contact with them. So this is the whole thing with the mask debate when it comes to COVID. People say, "Oh well, it's not there to protect me from it's not." The masks aren't able to filter out viruses. No, they're not able to filter out viruses. That's true. That is a valid point. However, the masks do filter out respiratory droplets. And a virus can't just jump into the air and find its way to your mask and just wiggle itself through on its own. It has to be attached to something. So, the reason we use masks now is because it prevents creating airborne respiratory droplets. So, if somebody is wearing a mask, myself and the person in front of me is, I'm only wearing a mask and the person in front of me is not, and they sneeze, right? Since I have the mask on, the respiratory droplets should be filtered by that mask. It should block the respiratory droplets from getting to my respiratory system, my nose and my mouth. If neither of us are wearing masks, then we're creating these respiratory droplets that both of us can pass back and forth. And if we are both wearing masks, then there's very little chance of transmitting the pathogen from one person to the other through respiratory droplets, because the mask catches those respiratory droplets. So when you're wearing a mask, you're doing it to prevent respiratory droplets, airborne droplets. Not because they filter out pathogens. They're not that good in terms of the viruses are too small, but they block respiratory droplets, which viruses are attached to. So, in the end, it actually works out that masks are extremely efficient in preventing transmission. So, wear a mask, wear a mask, wear a mask. It's not just for you, it's for other people. And this is one of the great quote unquote sacrifices we have to make as humans, is be compassionate for other people. Some people are extremely high risk for this disease. Extremely high risk. I'm one of them. I have a genetic heart condition, and I really should not be going to Walmart or anything like that. I don't, because people don't wear their masks, which puts me at a higher risk of getting infection and getting sick and possibly dying. So Um, if there's one thing I can kind of stress about this, is just wear your mask. It's honestly a small inconvenience in comparison to the safety of the population. This is very, very small inconvenience. And we'll talk more about COVID later. So now I started to talk about these stages before, or these definitions before I move back to that slide. There are 5 general stages of disease, and there are some definitions that we need to clarify before we move on to those stages. So first off, infectivity. Infectivity tells us how good of an infectious agent it is at establishing infection. So something with a higher infectivity is really good at establishing an infection. Shigella needs, let's say, about 10 to 15 cells to cause an infection, meaning it has high infectivity. Virulence describes the severity of disease following the infection. So you can say a certain pathogen is extremely virulent, meaning it causes severe illness. Again, Shigella. Shigella causes dysentery, which is diarrhea, bloody diarrhea, mucus in the stool, and some more pretty serious signs and symptoms. So we could say Shigella has a high virulence. It's pretty severe. And then pathogenicity is the ability to cause disease. So we've been using the term pathogen this whole semester. So pathogenicity is an organism's ability to be a pathogen. So some organisms like E. coli, the ones that you work with in lab or in your lab kit, they're non-pathogenic, meaning they don't have pathogenic genes. However, some organisms can have pathogenic traits. So the ability and have the ability to cause disease. Here are the 5 stages. One is incubation. 2 is prodromal. 3 is acute. 4 is period of decline. 5 is convalescent stage. So what this means is incubation period, stage 1. This is when the pathogen colonizes let's say your body, and it starts to replicate and increase in numbers, and you start to see maybe ever so slight symptoms, ever so slight. But during incubation, that's where you're getting lots of replication, lots of trying to colonize your body. You get to stage 2, the prodromal phase. This is where you start to feel early symptoms. So if you're sick, you know that probably if you've ever had the flu, even before you feel extremely sick, you start to feel slightly sick beforehand. You feel a little bit nauseous. You have a little bit of a headache. You're a little extra tired, just ever so slightly. So those early symptoms are what we call the prodromal phase. Then you reach the acute phase, and this is where the disease is extremely virulent. And basically it's the stage that the disease peaks. Meaning, and what I mean by peaks is that it's excuse me, the signs and symptoms are the most obvious during this stage. So flu, for instance, after you get past the prodromal period and you move into the acute phase, then you're looking at, oh man, I'm throwing up. I am nauseous, extremely nauseous. I am not feeling good in general. I have a major headache that won't go away. So the more serious signs and symptoms during the acute phase. The period of decline is basically when the body starts to regain control and starting to get rid of the pathogen, and you start to see the symptoms starting to go away. Doesn't mean you're completely better. It just means you're starting to feel better. And that's probably because you've taken an antibiotic, or you've gotten a steroid shot, or sometimes you just need your immune system to catch up and take care of it. And then you move into 5, the convalescent stage. This is where you are fully recovered. And in some cases, the pathogen remains in the body, but it goes into a latent stage. Remember, we talked about latent diseases where they can hide for years, possibly. So you get to phase 5, convalescent stage, and all of a sudden you're looking at a disease that you've recovered from, but sometimes the pathogen is kept latent in the patient. Now, hopefully you understand that we're looking at time on the x-axis and then the level of pathogen on the y, and these 5 stages ring true for almost all organisms, almost all pathogens, but the distribution is different. They don't all fall perfectly into this curve right here. The acute phase could be shorter, or the prodromal phase could be longer. So this is just showing you in general roughly what it should be, but it does not mean that all of them follow this exact curve. In fact, most of them don't. The phases are actually different depending upon the pathogen. Some pathogens, you develop symptoms almost immediately. If you have food intoxication, you get sick within possibly 4 hours, 4 to 6 hours, which is very different than tuberculosis, where it takes, the generation time is roughly 18 to 24 hours. So the incubation period is going to be much, 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 much longer than something that causes an acute disease. Point is yes, they follow the stages. No, the curve is not the same for each organism. All right. Moving into epidemiology. So epidemiology is basically focusing on diseases, both infectious and non-infectious, in populations in order to understand them, prevent them, and track and monitor them. And epidemiologists do all of those things. There are so many functions that epidemiologists have that it's hard to really say, okay, an epidemiologist does X. Well, sure, they probably do X, but they also do Y, Z, A, B, C, right? They don't just do one thing. They can track diseases. They can estimate transmission rates. They can they're really important in terms of getting literature out and preventing illness in communities, making people aware of specific diseases. So it is important that we understand epidemiology, study of what is upon the people, encompasses a lot of kind of subsections. And here we combine them into two general goals, but you can see how there's a lot to those two goals. First one, describe the nature, cause, extent of the new or existing disease in the population. And two, intervene to protect and improve health in populations. So both of these fall under the scope of epidemiology, but you can see how there are multiple sections to this. We group them into two major goals, but in the first one, describing the nature, describing the cause, the extent, tracking populations, all of that is extremely important and falls under the scope of epidemiologists. Now, in epidemiology, they have the epidemiological triangle, and the epidemiological triangle basically is a Venn diagram where you have three distinct factors that are separate, but once you put them together and they overlap, that's where you get your disease. So it's basically saying that there are numerous factors that are associated with the cause of disease. So for instance, environmental factors, location, global location or geographical location, water source, food source, climate. Host factors, are you healthy? What's your lifestyle like? Are you in a high-risk occupation? And then on the third one, it's the etiological agent, specifically the organism that is causing the disease. So all of these things have to come together and overlap somewhere, and that's where you get your disease. And you can see right here, where all three of these overlap, that's where you see disease. Now, looking at all of these, all three of these, environmental factors, host factors, and etiological agents, there's a lot of things we can take away from this. Environmental factors, geographical location, or host. Let's say you live in a developing country, a country that is extremely underdeveloped. You may have poor access to fresh water. You may have lots of pollution, which can lead to problems. Host factors, age, the older or the younger you are, really young, really old, more susceptible you are. Are you in good health in general? Do you have genetic predisposition for diseases? And then the etiological agent, the bacteria or the virus or the fungi, essentially that organism or the organism is part of this triangle. And where they all overlap is where you get disease. So in some cases, knowing the environmental factors and host may be more important than knowing the causative agent, which is really interesting because if you know certain environmental factors cause certain and increase the risk of certain pathogens, then you know that there's a list of organisms that it could potentially be that's causing the disease. For instance, listeria. It's found in rat pee and mouse pee. Basically, what happens is in the sewer systems, mice, rats pee, listeria gets into the sewer systems, and when there's flooding, the sewer systems are backed up, the water comes up, and now the water is contaminated above the sewers, and if there's a person that's trying to escape the water, right, they're trapped, and they have to wade through the water to get out of there, they're potentially coming in contact with extremely dangerous pathogens. So, does it matter that it's listeria? Not really. Okay, environmental factors are extremely important. So, a great example of this would be Hurricane Katrina. When Hurricane Katrina hit, and we'll focus on New Orleans, when Katrina hit and New Orleans was essentially underwater, all these sewers that were perfectly fine beforehand started to flood, and all that water, the sewage water, came up and was above ground. And people who were trying to escape that particular area had to wade through the water, some of them, and had to get away. But what we saw was, afterwards, we saw numerous cases of specific diseases associated with environmental factors, like coming in contact with contaminated water sources. So we may not have known that it was listeria at the time, but we knew that, well, the flooding led to contaminated waters, people came in contact with the contaminated water, and then also the host, that person, may have been more susceptible to getting sick than somebody else. So knowing the environmental factors in the host may sometimes be more important than knowing the actual organism. Now, granted, to treat somebody, you need to know the etiological agent, the organism that causes the disease. But sometimes knowing the other two first is just as important. This is where I'm going to end the first video of Chapter 9. I'm going to post this, and the second video will come out in the next 24 hours or so. I wanted to break this up into two videos because the material is a bit dense. So this is the end of video 1. Video 2 will be posted shortly.